close one eye. With the other, look at something across the room. What you're doing right now, the light entering through a transparent lens, bending, hitting 120 million photoreceptor cells arranged on a curved surface at the back of a fluid-filled sphere, converting photons into electrical signals that travel down 1 million nerve fibers to a visual cortex that assembles all of it into a coherent image in real time, is one of the most sophisticated optical instruments ever built. Darwin looked at the human eye in 1859 and called it a problem, not because it was poorly designed, because it was too good. In On the Origin of Species, he wrote that the idea of the eye having been formed by natural selection seemed, he freely confessed, absurd in the highest possible degree. He spent 3 paragraphs defending himself. He was not comfortable with that section of the book. He was right to be uncomfortable, but not for the reason he thought. The problem with the eye is not that it is too complex to have evolved. The problem is that it evolved too many times. Not once, not twice. Depending on how you count, the eye has arisen independently somewhere between 40 and 96 separate times across the tree of life. Each time, starting from nothing. Each time producing something that could detect light, and each time arriving at a completely different solution to the same problem. This is not a story about a single invention. It is a story about the universe having a limited number of ways to turn light into information, and life finding every single one of them. The first light-detecting structure in evolutionary history was not an eye. It was a patch of photosensitive cells on the surface of a simple organism, not much more than a cluster of proteins that change shape when a photon hit them. Useless for forming images, useful for one thing only: distinguishing light from dark, day from night, shadow from open water. That is enough. A creature that can tell when a larger animal passes overhead, casting a shadow, has a survival advantage over one that cannot. Natural selection does not require perfection. It requires better than what came before. From this simple patch, the logic of improvement runs in one direction: more information about where the light is coming from. A flat patch tells you light exists. A curved patch, cells arranged in a slight concavity, begins to tell you the direction of the light source. Curve it further, and the directional resolution improves. Add a small opening at the top, and you have a pinhole camera. The same principle as a pinhole photograph, focusing light onto the back wall of the depression without any lens at all. This design exists today in the Nautilus, a cephalopod whose lineage extends back nearly 500 million years, a survivor of every mass extinction since the Cambrian. The Nautilus has no lens, it has a pinhole. Its vision is blurry, limited in resolution, but it works. It has worked for 500 million years without requiring any improvement. Evolution, given time, kept going. A transparent membrane across the pinhole opening keeps out debris without blocking light. Fluid fills the chamber. Then, under selection for sharper images, the refractive index of that fluid begins to vary. Denser at the center, less dense at the edges, and a lens forms. Cells at the center of the membrane become more transparent, more curved, more optically precise. In 300 million years, roughly, working in geological time, accumulating small improvements, a patch of light-sensitive skin becomes a camera eye. The vertebrate eye is a camera, a single large lens, a variable aperture, a curved photoreceptive surface at the back. So is the eye of the octopus. Both are optically sophisticated. Both work on the same principle. Both have a lens, a retina, an iris, a vitreous chamber. They are not related. The vertebrate eye and the cephalopod eye evolved completely independently, separated by hundreds of millions of years and an entirely different branch of the tree of life. Yet they arrived at the same architecture. There is one notable difference. In the vertebrate eye, the photoreceptors point backward, away from the incoming light, and the nerve fibers pass through the retina before collecting at the optic nerve, creating a blind spot. In the octopus, the photoreceptors face forward, correctly oriented toward the light. No blind spot. The octopus, evolving independently, solved the wiring problem that vertebrate eyes never did. But the camera eye is only one solution. Evolution found others. The compound eye, used by insects, crustaceans, and many arthropods, takes the opposite approach. Instead of one large lens forming one image, it uses thousands of tiny lenses, each pointing in a slightly different direction, each capturing a small portion of the visual field. The brain assembles them into a mosaic. A dragonfly has 28,000 individual lenses in each eye, together covering nearly 360 degrees. It cannot focus the way a vertebrate can. The resolution of a compound eye is limited by the number and size of individual facets, but it detects motion with extraordinary precision. A dragonfly intercepts prey in mid-air with a success rate of over 95%, not by seeing a sharp image, but by tracking motion across thousands of simultaneous points of data. The compound eye and the camera eye evolved from the same ancient light-sensitive patch. They diverged before the Cambrian explosion more than 540 million years ago and have been running as parallel experiments ever since. Then there is the Mantis shrimp. The Mantis shrimp has 16 types of photoreceptors. For comparison, humans have 3, sensitive to red, green, and blue. Most mammals have 2. The Mantis shrimp, living in shallow tropical reef environments, processes 16 distinct wavelength channels simultaneously, including ultraviolet and infrared light, completely invisible to any vertebrate. For years, scientists assumed this meant Mantis shrimp experienced a visual world of incomprehensible richness, that they saw colors humans could not imagine. Then behavioral experiments revealed something stranger. Mantis shrimp are worse at distinguishing between similar colors than humans. They don't use their 16 channels to discriminate finely between wavelengths. They use them as a rapid parallel identification system, each wavelength channel acting like a binary detector. The visual system of the Mantis shrimp is not a high-resolution color camera. It is a high-speed parallel scanner, an eye built for a completely different computational purpose. The scallop, a bivalve mollusk, a shellfish, has up to 200 eyes arranged along the rim of its mantle. Each eye is roughly the size of a pinhead, and each one works on a principle found nowhere else in biology. The scallop eye has no refracting lens. Instead, it uses a mirror, a layer of precisely arranged guanine crystals, the same molecule found in DNA, lines the back of each eye, reflecting light onto 2 separate retinas positioned in front of it. The scallop eye is a reflecting telescope in miniature, using the same optical principle as the Hubble Space Telescope. It evolved this solution independently, in a creature with no brain to speak of, to detect approaching shadows and trigger a reflex that snaps the shell shut. 200 reflecting telescope eyes on a shellfish. This is what evolution does when it has enough time and no constraints. The box jellyfish may be the strangest case of all. It has 24 eyes, arranged in 4 clusters of 6. 14 of the 24 are simple, pit eyes and slit eyes detecting light and direction. But 4 of them, one in each cluster, are camera eyes, a cornea, a lens, a retina. In an animal with no brain, no central nervous system, no cortex to process the image, just a diffuse nerve net and 4 camera eyes pointing outward into the reef. How it uses the image is still not fully understood. All of these independent inventions, the camera eye of vertebrates, the camera eye of cephalopods, the compound eye of arthropods, the mirror eye of scallops, the pinhole eye of the Nautilus, the multi-lens eye of the box jellyfish, share almost no structural relationship. They are built from different tissues using different optical principles wired to different nervous systems, and yet they are all controlled by the same gene. PAX6 is a master regulatory gene, a genetic switch that initiates eye development in almost every sighted animal on Earth. It is active in the developing eyes of humans, flies, octopuses, mice, squids, jellyfish, and ribbon worms. Expressed in the eye of a mouse, it can trigger eye development in a fly, building a fly compound eye on a mouse. It is so conserved across 600 million years of separate evolution that the instructions it carries are still compatible across phyla that have been genetically diverging since before the Cambrian. Natural selection discovered multiple optical solutions, but it always reached for the same master switch to begin building, which means the eye is not one thing. It is a family of inventions all solving the same problem: how to turn light into information with different tools, different architectures, different computational strategies. And it means that wherever on Earth evolution had access to light and time, it built something to see with. Every time, without exception. The question that follows is one scientist rarely asked directly: if the eye evolved independently 40 to 96 times on a single planet, using the same physical laws that govern every world in this universe, what are the odds that vision has only ever appeared here? Light travels at the same speed everywhere. Photons interact with matter the same way on every planet in every galaxy. The physics that made the eye inevitable on Earth does not change. Evolution has not invented vision. It has discovered it, over and over and over again, and it is almost certainly not done